now the next portion under transcription is mechanism or steps in transcription. Simple ആണ്. നമ്മൾ ഇതിനു മുന്നേ പഠിച്ച ടോപ്പിക്കിൽ പഠിച്ചത് the three regions of transcription unit. ഇതിൽ the three steps in the process of transcription. That is initiation, elongation and termination. Initiation നടക്കുമ്പോൾ എന്താണ് സംഭവിക്കുന്നത്? During initiation what happens is that we know that there are three regions. For the transcription unit there are three regions. So this is one region, this is another region, and middle one is one region. So this is the 3-5' strand. This is the 5-3' strand. So the strand with 3-5' polarity is acting as the template strand or anti-sense strand. The other strand is displaced and that strand is called as a sense strand or the coding strand. So anyway, this strand is important. Now, at the time of initiation, what happens is that RNA polymerase enzyme and some protein factors which are needed for transcription called as transcription factors comes and binds to this region. So that is the first step happening during transcription, that is initiation. But during initiation what happens is that the RNA polymerase enzyme along with certain protein factors which are needed for transcription comes and binds to the promoter site. In case of eukaryotes, we just name them as transcription factors. But in case of prokaryotes, they have certain specific name. They are named as sigma factors. So sigma factors and RNA polymerase will bind to the promoter site in case of prokaryotes. In case of eukaryotes, RNA polymerase and certain transcription factors binds to the promoter site. Then, second step is elongation. What is happening during elongation? During elongation, the RNA polymerase will be moving like this. That time, RNA polymerase will polycatalyze the polymerization of ribonucleotides complementary to that present in the DNA, and a new mRNA or the RNA is formed. So, elongation process lana structural gene copied down. So, structural gene lulla nucleotides na complementary ആയിട്ടുള്ള ribonucleotides ne join ചെയ്ത് അതാണ് polymerize ചെയ്തിട്ട് mRNA is or an RNA is formed. That is called as elongation. So when the RNA polymerase reaches the terminator site or the terminator sequence, what will happen? The RNA polymerase will separate off. In case of eukaryotes, it will get separated off and the newly formed or nascent RNA will get separated from the DNA strand. So that is what is happening in case of eukaryotes. In case of prokaryotes, there are specific proteins called as Rho factors. So Rho factors will come and bind to this region, stopping the process further. So that is called as termination. This is a stoppage of the process of transcription. So itreyullu. So this is the mechanism or steps in transcription. Very simple. 3 steps are there: initiation, elongation, termination. Initiation നടക്കുമ്പോൾ ഓർക്ക് RNA polymerase and the protein factors are binding to the promoter. Elongation നടക്കുമ്പോൾ the structural gene is copied by the RNA polymerase enzyme which is acting as or helping the catalysis for the polymerization of ribonucleotides to form the RNA. When it reaches the terminator sequence, what will happen? In case of prokaryotes, Rho factors will come and bind. In case of eukaryotes, the RNA polymerase will separate and the newly formed RNA is getting separated. So it's very important for the exam. It's a very easy topic also. So these are the 3 things happening during in the steps in transcription.